All right, how are we all doing? Am I muted? We're good. Did I remember to record? Yep, we're recording. All right. It's funny, I just sat down and my whole desk is cluttered with these Nightmare Before Christmas little toys. My son's become obsessed, so we've been putting the DVD on him like multiple times a night, and as soon as it ends, he just wants to watch it again. So really into it. So today we're going to be talking about the central nervous system. So we're going to be talking about things like the brain and the spine, and we're going to talk about a little bit about protection of the central nervous system as well. You will learn a more in-depth version of all this material when we do get into that section, whenever you happen to have your block for anatomy and physiology. So let's see here. Pause that. But with all of these, you're going to have just a regular list of structures like normal this time. You can look those all up. These, again, this is where your Complete Anatomy app is really going to shine, because you can go in and these deep structures, you can just pull that brain apart, get in there, rotate, and see what you need to see. So while you don't need to know the function of all these structures, it really does help kind of understand how it all fits together. And then again, we will be seeing it again in lecture. So be good. It's always good to see things a couple times to really understand them. So I'm going to kick off here and start sharing my PowerPoint. And then as always, if you're having issues with Complete Anatomy, we can tackle that at the end. I'm going to do screen share. All right, central nervous system. So here we have an external view of the parts of the central nervous system. So we can see that the CNS is organized in different components. We have the spinal cord, okay? We can see that at this top structure right here. I'll switch to pen. Okay. The spinal cord, that's going to run through the foramen of the vertebrae, the vertebral foramen, and that's going to help that bony structure of the vertebra adds protection to that spinal cord. Then we have the brain stem. Let me see here. Spinal cord, brain stem, and you can see a lot of that. The entire structure is internal there. So we can see midbrain, pons, medulla, and that midbrain then is internal only. That's the only way we can see that is on this cross section here. What's next? Then we got the cerebellum at the back of the brain. The diencephalon, which we can see on this internal view here. And that's comprised of the thalamus and the hypothalamus. At least those that have had lecture, we've talked about those structures. And then we have the cerebrum. And the cerebrum makes up the majority of the mass of our brains, what we tend to think of when we think of the brain. And that's comprised of that outer area, the really convoluted area with all the gyri, those folds. That's called the cortex. And so that's the outer folded area. And then there's the inner folded area too, right? So this is part of your cerebrum as well. I should point out that these structures, some of them have what we call bilateral symmetry, and that just means there's a left and a right mirror image of each other. So for example, if we were to look at the thalamus, that's going to have what we call bilateral symmetry. Same with the hemispheres, right? There are great models for these that are in that 110 that's been open for you. What we do ask with these, if you pull this model apart, it's a great way to look at it and see how it all fits together, these different structures, but please put it back together. Otherwise, it takes the TAs quite a long time to go in there and try to piece this back together, even though they know it pretty well. You know, it's still like doing a big puzzle every time. The other thing, of course, we've already talked about is please leave them in that room. Don't take them back to your dorm or there'll be a witch hunt. So let's look at the basic pattern of organization within the central nervous system. So we can see some consistent patterns here if we move through cross sections of these different regions, right? So if we move here, if we're looking at CNS here, or CNS here, we'll notice some general trends. And what we see is that within all sections of the CNS, both upper and lower, there is a central cavity. Blue probably wasn't the best choice for the arrow, but there's a central cavity. It's very small there within the spinal cord. And that's surrounded by gray matter. Okay, we'll go into the difference between gray and white matter really quickly, shortly, I should say. So you have a central cavity, and that's surrounded by gray matter. And that central cavity, it's not just hollow, right? That's going to be filled with cerebrospinal fluid. Okay? Helps add protection, moves nutrients in and out the cerebrospinal fluid. So within this surrounding gray matter here, we see that in all three of these cross sections. Let me pull up chat here. So that I can see. I'd like to get a response from some of you. Do you think that that is going to be cell bodies or axons of those cells? So we're talking about gray matter that's surrounding that central cavity. Someone can take a stab at this, type in the chat. Thank you. Thanks, Matt. That's right. That is going to be the cell bodies. Okay. So they're darker, and we can think of these as the computational centers of the brain, those cell bodies. Okay. And then next to that, you can see we have this ring of outer white matter then within all these different sections of the CNS. Okay. And these are white due to the myelin that's running along those axons. So they're myelinated, appears white. And we can think of those axons, that white matter area as the information highway. So this is where we're going to be transporting info to that gray matter, the cell bodies, via those long white axons. Okay. And then finally, what we see on this upper portion, so the upper CNS, we see an additional outer layer of gray matter. And that's what we call the cortex. And that's where the higher levels of thinking are done within our higher species, like humans. I think that's good for that one. Okay. The top left, we got the brain cross section there. And again, we can see this pattern going from gray to white to gray. And first we have that outer cortex here. So like the arrow, a convoluted area. So those are those cell bodies. And then deep to that, we have the white matter again. And then within the brain here, we have that inner layer of gray matter. You can see this on both sides here. And we call that the cerebral nuclei. So these are clustering of cell bodies within the brain. And we call them nuclei within the CNS. And this isn't like the nucleus of a cell, okay? Keep that in mind. It's a clustering of cell bodies when we're talking about the central nervous system. Also note that that clustering of cell bodies within the peripheral nervous system would be called ganglia or a ganglion. Okay? Here we're talking about a nucleus or nuclei when we're talking about the CNS, just a bunch of cell bodies that form a cluster of gray matter within the brain. We can see that the two hemispheres of the brain are connected by the corpus callosum. So those are just, again, that's white matter, so that's axons that are running back and forth between the two hemispheres so that they can communicate with one another. And over here, just so you can see what we're talking about here, you can see how that neuronal cell body would give rise to gray matter. And then the axon, with those lipid sheaths that are wrapped around them for insulation, that would give rise to the white appearance. So that's why we have that gray to white to gray again with these nuclei. Okay. So now let's talk about brain support and the meninges. So this is kind of a detailed picture here, but I don't know what I want to point out here. Well, I'll just skip to the next slide and work through this slide individually. So we got some brain protection, and the first layer of protection we call the cranial meninges. So these meninges are like different layers. Okay? And it's 3 connective tissue layers that make up the cranial meninges. Okay? And what they do is they separate and support the soft tissue of the brain. So they surround the brain, these 3 layers. They enclose it, they protect it, and they protect the blood vessels that supply the brain as well. And also, something we're going to talk about, an interesting role, is within this hollow space they create right here, they're going to contain, awesome, and circulate the cerebrospinal fluid, or we'll just abbreviate that, CSF. So we can see, if this is the brain right here, the innermost layer is going to be called the pia mater, okay? And that adheres directly to the brain. It's kind of a weird translation. That actually translates to tender mother, okay? Mater and pia being tender. And this is this thin, delicate layer of areolar connective tissue, so the tender mother. Mother maybe being a protective type thing with these protective layers, right? So innermost is the pia. Let's do a 1. Next, we have the arachnoid matter. Okay? And you can remember this one because with this layer, we have these arachnoid trabeculae. Okay? And what it looks like is kind of these web formations that would be formed by a spider, arachnid. Okay? So the arachnoid matter is filled with these trabeculae, and these trabeculae are made up of like collagen and elastin. And they create this hollow space for the cerebrospinal fluid to circulate through. It also helps form a protective layer then, like a cushion then. Okay. So this creates the, I don't have it on there, but this is going to create your subarachnoid space. Maybe I'll just draw that here. So this is your subarachnoid space within the arachnoid mater that's formed by these trabeculae of the arachnoid mater, the spider layer. Okay. That's going to fill with cerebrospinal fluid. Then the outermost layer, then we have the dura mater. And dura, as you can guess, that means hard or tough. And it's made up of these two fused layers. And in some areas, those layers are actually going to split and come apart and form what we call a dural venous sinus. So we're going to form a cavity or a sinus. And we'll work through why that's important in a little bit. Basically, it helps to drain excess cerebral spinal fluid that would build up within the arachnoid space. So here we can see all of those layers put together now. So we have the innermost layer. You can see it's this really thin pink layer adhering directly to the brain. That's our pia mater. And then we have that arachnoid mater external to it right here, creating that arachnoid space. And we can see those trabeculae really there, that web-like structure of collagen and elastin. And then that outermost layer, again, is the dura mater, which is actually two different layers. I don't believe you need to know that. So just remember, it's two fused layers that make up the hard outer layer, the dura mater. And then as I mentioned, in some cases, we get splitting of that dura mater. We can see there's a layer here and a layer here, okay, of the two different layers of the dura mater. And you can see that they split in this case, and they open to create this sinus, okay? And within this sinus, you get these little projections of the arachnoid layer that we call an arachnoid villus, okay? So let's move on. We'll see why that's important in regards to cerebrospinal fluid. How then do we prevent the accumulation of CSF so that we do not have cerebral edema? Well, we need some way to get rid of excess CSF. And what occurs is that excess CSF is going to flow into what we call arachnoid villi. So kind of protruding from that subarachnoid space, we have a villus that appears, and that helps drain excess CSF into the dural venous sinuses. Now, I'm going to show you exactly how that works on the next slide. So first, you got the brain here. And so I'll just do kind of those convolutions, the gyri of the brain. And then atop that, we'll have our first layer, the pia mater. We'll put the pia mater directly atop the, remember it adheres directly to the surface of the brain. Okay? And then above that, then, we are going to have our arachnoid layer of the meninges. And associated with that, we have this little arachnoid villus that projects up into the dural sinus. Beneath that, we're going to have our subarachnoid space with those trabeculae in there, just collagen and elastin. Okay. And this is where, remember, our CSF flows. Okay. So some of that CSF is going to enter up into this arachnoid villus. Okay. But then we have our last layer that we need to draw, and that is the dura mater. And remember, the dura mater is actually two separate layers. So while they're normally together, when we get to this villus here, they actually will split to give rise to that dural sinus. Okay. So this is that dural sinus here. That says sinus. Apology, my tablet hasn't arrived yet. So that excess CSF is going to be pushed here through this arachnoid villus, and it's going to enter the dural sinus, where it gets a chance to return to venous blood supply. And that's it. Okay, that was a drawing that I had pulled from the lecture and put in there. That's why I was kind of talking over myself there briefly. That's how we drain excess CSF. We also see that these meninges, that they're going to separate, we said that they separate and support the soft tissues of the brain. So one we can focus on here is the falx cerebri, or cerebri. It's also known as the cerebral falx, and it's named for its sickle-like form. So you can see this falx runs down deep between the hemispheres. This is actually all falx cerebri here, but it makes this sickle-like shape. If you don't know what a sickle is, it's kind of what like the reaper carries, right? The blade used to scythe, cut down wheat or whatever they'd cut down with it. And so it makes that arching falx sickle-like shape there. Okay, so it's a large crescent-shaped fold of that meningeal layer of the dura mater, and that descends vertically into the longitudinal fissure between those two hemispheres. So here we have our longitudinal fissure, and it just separates them. Okay, so it separates the cerebral hemispheres of the human brain, that falx cerebri. There are two other areas where we get separation from these meningeal layers. We have the tentorium cerebelli. Okay, so that cerebelli should be some type of keyword because it sounds like cerebellum, right? And this separates both the occipital and temporal lobes from the cerebellum. We'll look at the lobe shortly, so this will make a little bit more sense. And then finally, we have what we call the falx cerebelli, not the falx cerebri. Okay. And the falx cerebelli, as the name kind of says, divides the halves of the cerebellum, that back portion of the brain, that lower back portion. Now we also have these, when we're talking about brain protection here, we also have these cavities within the brain that we call ventricles, okay? And they're not hollow areas of the brain. If you were to go into lab, into 110 there and take apart the brain model, it's gonna be represented within most of those models, except one that I can think of as just a hollow area. It's actually not hollow. Those ventricles are gonna be filled with, again, cerebral spinal fluid. And the brain kind of then is supported by this and just floats atop that ventricles, those ventricles, I should say, okay? So those ventricles, they're not hollow. They're actually lined with areas of concentrated ependymal cells that secrete cerebral spinal fluid. Okay? And they're actually all connected, these different ventricles, and the CSF flows through them. Okay? So in fact, the CSF, remember we said it's found within that subarachnoid space? Well, it can actually move for these apertures, so into both the, to surround the spinal cord and into those, the arachnoid mater as a cushion of CSF that surrounds the brain as well, okay? So it's actually this median aperture here where on the fourth ventricle where it's gonna enter into that subarachnoid space of the meninges. There are four ventricles that we can see here. You got two lateral ventricles, so you can see them here, right? And here, lateral, because that's where they're found. They're found laterally. The other two ventricles are found medially. And you can think of these as ventricles 1 and 2, because the others are named third ventricle and fourth ventricle, but 1 and 2 they just decided to name lateral. Okay. So we have the third ventricle then, and then the fourth ventricle. So let me just show you this. So there's the lateral ventricles. They kind of look like wings, right? So start to see this pattern kind of appear. Then we have the third ventricle, and then we have this fourth ventricle. The way I like to remember this, how this is positioned there within the brain, because these kind of look like the wings of a dragon that are sticking out. The body then of that dragon would be this third ventricle, and the tail that's hanging down and trailing would be the fourth ventricle. So you can kind of superimpose this dragon-like shape on top of it, okay? All right, so let's talk about the spinal cord then. The spinal cord is a long, slender cylinder of nerves. And it actually extends from the brainstem here down to between about the first and second lumbar vertebrae. Okay. So all in all, it's about 18 inches long. It's about 45 centimeters. And it has a 2 centimeter diameter, like 3/4 of an inch, I think, is the translation for that. It extends through the vertebral canal, the spinal cord, and it's connected to spinal nerves. So we can see these paired spinal nerves that come off of the spinal cord, and those are protected by the projections of the vertebrae. Okay. I should also point out that we still have those 3 layers of the meninges that we talked about within the brain. Those are also found here within the spinal cord. So they have that CSF to help move nutrients and that little protection. And I think you're familiar with this because a lot of you have probably heard of getting an epidural when you're pregnant and about to deliver. Okay? And that's delivered in the epidural space. So they'll take that needle, they'll have you flex your back so they can get in between the vertebrae, and they will insert that into epidural space. And that's between those 2 layers of the dura mater is where they inject that analgesic. Okay? All right, so here we can see those alternating gray and white layers. Again, the gray and white matter, but here we're in the spinal cord now and not in the brain. So we lose that outer gray matter, the cortex that we saw within the brain. We got that central canal surrounded by gray matter and then surrounded by white matter. That's the pattern. And you'll also notice that within, the computer's just doing stuff on its own here. Hold up. I need to get rid of my timings on this slide. That's what's screwing everything up. So we'll do slideshow, clear, timings on all slides. There you go. It's progressing without me wanting to progress. So what we see here is we have these different horns there that the gray matter or those neuronal cell bodies make up. And each of those horns of the spinal cord gray matter there houses a different type of neuronal cell body. So we have 3 different horns we can see. We see the posterior, my pen's gone. You can see the posterior horn, the lateral horn, and the anterior horn. And you'll see they form, yeah, that's also called the dorsal. You'll see that within your text. They'll call the posterior horn the dorsal horn, or the anterior horn the ventral horn. They're the same thing. Okay? And you'll see they form this butterfly-like shape, right? So this will be your posterior horn, and this would be your anterior horn, and the lateral horn somewhere within the middle. So it forms a butterfly pattern. And then surrounding that, we can see that we have the white matter. And this is organized into bundles that we call tracts. So they run vertically, bundles of nerves running up to the CNS, bringing information to the CNS, or out from the CNS to the periphery. So it's just a bundle of axons, right? Well, let's talk about these different horns and what's going on there. So the different horns house different neuron cell bodies. We mentioned that. So if we look at the posterior horn here, remember that's that dorsal horn, you could call it, right? Like dorsal, like back, dorsal fin. Within that posterior horn, this is where we're going to see incoming information from sensory neurons. Okay, so incoming information from the periphery is going to enter here into that posterior horn. Now, if we look at the lateral and anterior horns, These are associated with outgoing messages. So the anterior actually is going to go to your skeletal muscle. And the lateral, that's your autonomic motor neurons. There's going to be things like going to smooth muscle, cardiac muscle, or different glands. Okay. So what you need to take away from this is within that back horn, things that enter the back of the spinal cord, that's incoming info. Well, it's kind of screwed that up. But whereas things that are coming out of the anterior and lateral horns, that's outgoing information. Okay. So moving on to different areas of the brain now, we'll move on to the brainstem. Okay. And this is comprised of the midbrain, the pons, and the infamous medulla oblongata. So any Waterboy fans in here know that the crocodiles aren't really ornery because they got all them teeth up in their skulls or something like that. I still remember that from high school. So the brainstem houses centers that control automatic processes within our body. So this is kind of like the survival center, and it controls our basic functions. That's what you need to take away for now. So this includes things like cardiovascular centers, respiratory centers, digestive control centers. So these things that occur automatically that we don't need to consciously think about are controlled from the brainstem. As far as anatomy, so anatomy-wise, I'll point out a couple different structures. Here, within the medulla, we can see we have these structures called the pyramids. You can see they're slightly pyramidal, and there are two of them, hence the pyramids. And then lateral to that, we have these structures called the olives, and thankfully they are olive shaped, so that'll help you recognize those within complete anatomy. What else do we got? Down here on the midbrain, we have these, well, you can see there are two superior colliculi, and then there are two inferior colliculi. And collectively, we call these then the corpora quadrigemina, which just is Latin for the four bodies. And these are just reflex centers for vision and hearing, but that's your superior colliculi and your inferior colliculi. The cerebellum, so again, that's going to be right here at the back of the brain, and this baseball-sized portion of the brain directly under the occipital lobe here. Okay. And it looks like a mini brain almost. And we have this structure here with inside these white tracks, remember those are axons, that we call arbor vitae. That's Latin for the tree of life, because in fact, this does kind of look like a tree, right? With its branches extending out here. That's your arbor vitae. And the cerebellum is what we associate with balance. And we'll learn a few of the more roles of the cerebellum when we get into it within your lecture when you get there. The vermis between the two halves of the cerebellar hemispheres, so the right and the left. And it's the midline of the cerebellum, and it just separates the cerebellum into two cerebellar hemispheres, the vermis. Another view of the cerebellum, there's that pronounced tree of life there. And that brings us to the diencephalon. So the diencephalon, again, we could only see it within that internal view. We can't see that externally. And importantly, it houses the thalamus and the hypothalamus. Okay, so we start with the thalamus. This is where, just a little FYI, all sensory input that's coming from the periphery on its way to the CNS, it's going to go through the thalamus. Okay, so it's like a pit stop on the way, the integration center kind of. on the way to the cortex of the brain. And then beneath that, denoted by its name hypo, meaning below, we have the hypothalamus. Okay, so we have the thalamus here, and then the hypothalamus beneath the thalamus. And the hypothalamus is an integration center for autonomic processes. So those processes that automatically happen within our body, such as regulating body temperature or thirst control center. I should also point out while we're here that technically the epithalamus is part of the diencephalon, but I don't believe you need to know that, but that includes something that you're familiar with, the pineal gland. Okay. And that's what secretes melatonin. So it's responsible for our sleep-wake cycle. Epithalamus. Epi meaning upon the thalamus here in the back. More views of the diencephalon here. Let's see if there's anything to point out. There's that pineal gland of the epithalamus. Here's your thalamus here, and your hypothalamus. I don't know, just another view. It'll be clear within complete anatomy. All right, that brings us to, I debated starting with this, but there's no great way to work through this. So we have our cerebral hemispheres here. There are 4 lobes, and these are divided by deep grooves that we call sulci, or singular would be a sulcus. So for example, here we have the central sulcus, these really deep grooves that separate these different lobes. So the first one we have here is the occipital lobe. This is where we have our visual cortex. Those are eyeballs. Okay. And then we have our temporal lobe. And this is where we have our auditory cortex, right? So you can think of near your temples here, right? Because we have our ears around here. Temporal lobe near the temple. Then we have the parietal lobe. Parietal, no good way to remember this. It actually means wall. It acts as a wall to separate occipital and frontal lobes. But the parietal lobe contains our areas for somatosensory reception. So when I say somatosensory, that prefix soma, that means body, right? Like a cell body is a soma. So this is somatosensory, body sensory. This includes things like touch. That's the area of the brain responsible for perceiving touch. And then we have our 4th lobe, the frontal lobe. This is responsible for things like voluntary movement. Excuse me, it contains our motor cortex, right, controlling skeletal muscle. Excuse me, as well as things like speaking and thought. Again, you'll learn more about those within your lecture component for this course. It's an actual view of the brain. We can see between the hemispheres here, we have the longitudinal fissure. We can see those other structures we talked about here. You can see the pons and the medulla oblongata. Here's our frontal lobe. Posterior view, good view of the cerebellar hemispheres here. Here's your vermis. Internal view of your cerebral hemispheres. Not much to point out here other than, I guess we got a real view of those basal nuclei now, that clustering of cell bodies within the brain. All right, so I mentioned the sulci that separate the different lobes. We can see that here represented by these dark dashed black lines separating the different lobes. There's one sulcus I want to talk about here in particular, and that is the central sulcus. Okay? So this is between this frontal lobe and the parietal lobe. Okay? So remember, this is our parietal lobe here. This is our frontal lobe. And we have the central sulcus dividing them. And two areas of note we have here is before this sulcus, we have what we call the precentral gyrus. Okay. Remember a gyrus or a gyri is just a high, deep fold. So these are all gyri over the surface of the brain. And this particular fold here that they're highlighting in purple is the precentral gyrus because it's before this central sulcus. Okay. And then notice here behind the central sulcus, we have what we call the post-central gyrus. So that just means post-central fold. And that's what we have here. This precentral gyrus, shown in this lavender, that's our primary motor cortex. That's how we control movement. Okay? That's what that region of the brain is associated with. Okay? And then this post-central gyrus, this area behind this central sulcus, that's what we call the primary somatosensory cortex. Remember, somatosensory, soma meaning body, so body sensory. So this is body sensory input, again, like touch. So these are the areas of the brain associated with controlling skeletal muscle and then perceiving things like touch around us. Somatosensory. Okay. Now, I don't know how many of you have heard of the homunculus, right? But this is the homunculus here. And that translates to little man. And the concept of the homunculus is that the size of each body part indicates the proportion of the cortex dedicated to that area. So for example, right off the bat, we see that this little homunculus, this little man, has gigantic hands and gigantic lips. Okay, so that would mean that you would have a large, let's say we're looking at the somatosensory cortex, so kind of like touching, right? Detecting via touch. It's how much portion of this cortex is dedicated to that sensory detection. So you can see that lips are really big, tongue is really big, and hands are really large. So these are sensitive areas, right? And then our precentral gyrus here is the motor cortex. And again, how much of that cortex is devoted to those areas? We have really large hands. Fine motor control, right? Again, fine motor control needed for things like speech, so lips and jaw, I guess, and tongue. You can see there's not much devoted to things like your forearm, right? Or your knee, okay? So not much brain tissue we can see is devoted for those non-complex movements of the motor cortex. So they don't have much cortex dedicated to them. So that's the idea of this homunculus, little FYI. Oh, internal view, cerebral hemispheres. Again, we can see the corpus callosum, which connects those two hemispheres, those axon tracks running back and forth between the two hemispheres. I don't really think I had too much else for you here. Actually, this isn't even in your PowerPoint, I believe, because we are going to move these cranial nerves here to when we do peripheral nervous system. So I have a little less to do for this portion this week. So I can just get rid of that. So that is all I got for today. So now I'll stop screen sharing and take any complete anatomy questions. All right. If anyone's got anything. questions, comments, concerns. Otherwise, I'll see you again this time next week, I suppose, if not in class before that for some of you. Sound good? All right. Take care, everyone.
Thank you.
Yeah, you're welcome. We'll see you next week.